Hello, this is Josh Patel back again with another biology video. Today we were gonna do all of chapter 17, and chapter 17 is the tree of life, so we're gonna basically learn about life today. So 17.1 is the Linnaean system of classification. Key concept here is organisms can be classified based on physical similarities. So Linnaean is just the scientist that made up this classification, and it's classification used all over the world by all scientists in every country used to classify organisms. So Linnaeus developed a science naming system still used today. Taxonomy is the science of naming and classifying organisms, so that's a key vocab we need to know. So taxonomy is classifying and classifying organisms. So white oak would be that in Latin, and that is what we use in our Linnaean system. We use Latin. So this is the Latin word for white oak, and everybody all over the world uses it, so it's universal. So a taxon is a group of organisms in a classification system. So it's just a little group in our classes. So binomial nomenclature is a two-part scientific naming system. So bi is two, and nomenclature is basically giving names. So it uses Latin. Scientific names always written in Latin, in italics. Two parts are the genus name and the species descriptor. So that those are the two parts. The first one is the genus, and the second is the species descriptor. So here we see two is by, name nomial, naming nomen. It's a nomenclature. Nomenclature right there, and the system is clature. So the first word is the genus, second word is the species, and this is also important. So in the binomial nomenclature, there's two names for each organism. The first one is its genus, the second one is its species. And genus is one of the ways that we organize animals. So we have this, we'll get to it, I'll explain later. A genus includes one or more physically similar species. Species is the same genus or through the thought to be closely related. So species is things that are thought to be closely related. Genus name is always capitalized, and that's another important thing to know. The genus is always capitalized. A species descriptor is the second part of the scientific name, and it's always lowercase. So the genus is always capitalized, and the species, which is the second part, is lowercase. Always follows genus name, never written alone. So Tyto alba. So Tyto would be its what? Would that be its genus or its species? And since it's capitalized first of all, and since it's first, we know it's the genus. And alba would be the species of this owl. So scientific names help scientists to communicate. Some species have very similar common names. Some species have many common names. So this helps scientists communicate because in America we would call, let's say, we would call a dog. And in another part of the world, like in India, they'll call it something else. So they need a common name to call these animals. So they decided to use Latin as their Linnaean system of classification. So this is just showing how things are named. Linnaeus classification system has 7 levels. So each level is included in the level above it. This is what I was talking about earlier where I was going to explain, but it's coming up later. So levels get increasingly specific from kingdom to species. So this is how they start classifying things. So they get increasingly specific. So they start broad and they get specific from kingdom to species. So here we have kingdoms, and at the bottom we have species. And there are many different levels that we don't have to memorize in regular biology, but we should know of them, and we will figure out in AP biology for sure. So we start with kingdom, and we go to phylum, class, order, family, genus, species. They're not that hard to remember in order, but and there's some acronyms you can look up to help memorize them. And I think one is King Philip came over for great spaghetti. So K is for king, P phylum Philip, C for king, over for great spaghetti. So that's an easy way to remember it. The Linnaean classification system has limitations, so it's not the best. Linnaeus taxonomy doesn't account for molecular evidence. The technology didn't exist during his time, and Linnaean system based only on physical similarities, so they have to do with how the animal's built and what characteristics it has. And so based on characteristics, we place them in specific categories in the 7 layers. So physical similarities are not always the result of close relationship. So we can have two things that are similar, but they're not related at all. And the Linnaeus system was trying to show which ones are closely related, so it's not the best system. Genetic similarities more accurately show evolutionary relationships. So here, I think it's called a red panda we have here. Okay, so 17.2 classification based on evolutionary relationships. So this is what we try to do. We try to group animals that evolve together because we know they're closely related. Key concept is modern classification is based on evolutionary relationships. So what we do now, our modern classification is based on evolution. So cladistics is classification based on common ancestry. And that's exactly what we want to do. Phylogeny is the evolutionary history of a group of species. Evidence from living species, fossil records, and molecular data, shown with branching tree diagrams. So it's like starts from one main thing and it branches out to many specific things. So this is probably older in history, and then this is coming recent, and these are different species here. And it shows how they branched off from the original species, probably due to isolation and the things we discussed in the evolution chapter. Cladistics is a common method to make evolutionary trees, classification based on common ancestry. So if we look at this, they all have a common ancestry of this main branch here, and they each split off. So A and B would have common ancestries along with C, D, and all of them together. Cladistics is a common method to make evolutionary trees. Okay. Species placed in order that they descend from common ancestries. So that's basically the main branch is the oldest, and then it gets younger and younger as it goes on. So a cladogram is an evolutionary tree made using cladistics. A clade is a group of species that shares a common ancestor. Okay, so this is one of our evolutionary trees. It shows here they all came from here, and then they slowly branched off from each other. And as we notice here, they each get like more different from each other as they branch off. Each species in a clade shares common traits with other ancestors. Each species in a clade has traits that have not changed, or that have changed. So derived characteristics are traits shared in different degrees by clade members, basis of arranging species in a cladogram. More closely related species share more divergent characters. So this is kind of basic stuff. Closer they are on the tree, the closer they're related. The farther away, the more different they are. And each evolution or each branch, they change somewhat. So it represents on cladogram as hash marks. We don't need to know that. So, but we do need to know what a cladogram looks like and what they're used for. So nodes represent the most recent common ancestor of a clade. Clades can be identified by snipping a branch under a node. So 4 limbs with digits here. Okay, so the node is basically where they branch off. It's the point where they change. And a clade is just the group at the very end. So molecular evidence reveals species relatedness. Molecular data may confirm classification based on physical similarities. So as we know, molecular data can help us show how closely things are related to each other. Molecular data may lead scientists to propose a new classification. So this is just showing before and after how things changed. This was basically using how things look, and this is probably using DNA. So 17.3 is molecular clocks. So key concept, molecular clocks provide clues to evolutionary history. Molecular clocks use mutations to estimate evolutionary time. Mutations add up at constant rate in related species. This rate is the ticking of the molecular clock. As more time passes, there will be more mutations, and more differences as well. Mutations add up at a fairly constant rate in the DNA species that evolve from a common ancestor. So it's saying mutations occur at a steady pace, basically, so we can track them. So 10 million years later, one mutation in each lineage. The DNA sequence from the two descendants species shows mutations that have accumulated. So the black ones are the mutations, and they're slowly changing and getting more different, basically. So scientists estimate mutation rates by linking molecular data in real time. An event known to separate species. The first appearance of a species in fossil record. So mitochondria, DNA, and ribosomal RNA provide two types of molecular clocks. Different molecules have different mutation rates. Higher rate better for studying close related species. Lower rate better for studying distinctly related species. And yet again, this isn't that important to understand or know. Mitochondrial DNA is used to study closely related species, but this is important, which we already know that DNA is used to study species and see how close they are. Mutation rate 10 times faster than nuclear DNA. Passed down unshifted from mother to offspring. So it's saying that we pass on the mutations and eventually become different species. Ribosomal RNA is used to study distantly related species. So many conservative regions. Lower mutation rate than most DNA. So RNA mutates slower than DNA because it's safer. Domains and kingdoms. So these are domain and kingdom are two of the 7 classifications in the Linnaean system. Key concept is the current tree of life has 3 domains. So classification is always a work in progress. The tree of life shows our most current understanding. New discoveries can lead to changing in classification. Until 1866, only 2 kingdoms, Animalia and Plantae, existed. So kingdoms are basically one of the most broadest classifications in the Linnaean system. 1866, a single-celled organism moved to kingdom Prostate. So prokaryotes moved into the kingdom Monera, and then fungi moved into its own kingdom. So now we have 5 or 6. And then kingdom Monera split into kingdoms Bacteria and Archaea. So we're basically just keep expanding as our knowledge increases. And okay, so the 3 domains in the tree of life are Bacteria, Archaea, and Eukarya. Domains are above the kingdom level. So domains are even more broader than kingdoms. Proposed by Carl Hose based on RNA studies of prokaryotes. Domain model more clearly shows prokaryotic diversity. Domain bacteria include prokaryotes in the kingdom Bacteria, one of the largest groups on Earth. Classification by shape, need for oxygen, and disease. This is just specifics on what it takes to be bacteria. So domain Archaea includes prokaryotes in the kingdom Archaea, and cell walls chemically different from bacteria. So they're a little different from bacteria. Differences discovered by studying RNA, known for living in extreme environments. So Archaea are basically bacteria that live in either very hot or very cold environments, and those are considered extreme environments. So they're basically extreme bacteria. And as we know, bacteria are classified for their shape and need for oxygen and disease caused. And they're the largest group. So That's what archaea is, and eukarya includes all eukaryotes, and eukaryotes are, as we know, complicated cells like us. Kingdom Prostia, Protista, I meant. Kingdom Planta, Kingdom Fungi, Kingdom Animalia. So these are all the kingdoms in the eukaryote domain. And as we know, domain is the highest in the, or the most broadest in the 7 classifications. Bacteria and archaea can be difficult to classify because they're very similar, but there are some differences. Transfer genes among themselves outside of reproduction blurs the line between species. So we don't have a very solid line between classifying different things. It's kind of blurred. More research needed to understand prokaryotes. So we need more research to find more differences and like a key that we can use to classify things. So that's the end of chapter 17, which is basically all about the Linnaean system and taxonomy. And so next time we will be doing chapter 18, and chapter 18 will be a little different because it's not going to be the full power notes, it's going to be a little different.